housekeeping. Emergency exits are just right here, and then where you came into today. If anyone needs to use the restroom, it's just right across the hallway. All right, without further ado, I'd like to introduce Dr. Tracy Melzer. And yeah, thanks, Kelly, that's all good. And we welcome you along.
Great, thanks, Ty. Kia ora, everyone. So you're getting a double dose of American accents. So I hope it will be intelligible at the very end. So yeah, thank you very much for having me here today. It's a pleasure to be here despite the weather. And what I wanted to do today was talk about the work that we're doing at the New Zealand Brain Research Institute. So the New Zealand Brain Research Institute, which is kind of a mouthful, which is why we call it NZBRI, is located here in Christchurch. Some of you will be aware of the work that we do there. But what I wanted to do today was talk about specifically the brain imaging component of our work. So looking at pictures of the brain, how do we do that? What can it tell us? How are we using these images in a number of different examples of our work? So without further ado, I'd like to get started. And the way I like to start things is to talk about some fruit. Not because I think fruit is more interesting than brains, because it's obviously not. But I think it does provide a nice example of what imaging can tell us. So there is this guy, Inside Insights is his blog. He is an MRI technologist in Canada, and he has spent a lot of time taking images of fruit with MRI. And he produces some very beautiful images, which is just a nice example of how we can take pictures of something and look on the inside without opening that thing up. Right? I don't have to open up the mandarin orange. I don't have to cut open the apple to look on the inside. And the same thing is true of brains. We don't need to open up brains. We can actually use MRI or brain imaging to look at what's happening inside the brain. So before we start looking at brains, let's look at some fruit. So this is an example of fruit. This is a really nice example of a mandarin orange here. So we can take a three-dimensional image with MRI, right? We can slice that any way, right? We can slice it this way, and it looks like that, or we can slice it, along the side, and it looks like that. So I do need a little bit of participation here. So first up is that particular picture. Does anybody have any idea what that is? Okay, I'll give you a hint. It is fruit. Any ideas?
Mandarin.
Mandarin, close.
Grapefruit.
Oh, close. Grapefruit is close. We're in the same family. That is a lemon. So the nice thing to point out here about these different types of images is that you get what we call contrast, different contrast. So you can see the flesh looks different than the seeds, which looks different than the skin. Right? And that's essentially what we're using in MRI to look at the brain, look at the different tissues which we see with the brain. So maybe slightly easier. That one is slightly easier. This one? Corn. And again, I'll just point out how nice these images look, and they can tell us about different parts of the fruit, veg, or brain. So for example, you can see the kernels. They look very different than the cob, which look very different than the leaves. Go through a few more here.
Broccoli.
This one catches people out because it's not. Exactly, it's called. Yep. That's exactly right. This is the apple, and this is, again, another nice example of the fact that we can acquire a three-dimensional image, and we can chop that image up however we want. We can chop it this way, and we can recreate that image, or we can slice it lengthwise and create that image. Just a few.
Kiwi fruit.
Oh, that kiwi fruit, it is a tomato. Actually, that was slightly more difficult. This one I think is actually quite beautiful. Artichoke, exactly.
Pineapple.
One of my children's favorites, pineapple.
Sunflower.
Well done. And one more. Any idea what that one is?
Onion.
An onion, exactly. So this one is really interesting as well because as he was taking this image, before he put the onion in the scanner, he dropped the onion, and what you're seeing there is a bruise. Right? That again highlights exactly what we want to use brain imaging to look at in the brain. We want to look at any potential damage on the healthy areas of the brain. And so as I said, we can do the same thing with brains that we can do with fruit. So we can put little people or big people in MR scanners, and we can create images which look like this. And again, this is, I'll get back to that idea of contrast, right, where you can look at this and you can see that the skin, the scalp, looks very different than in the brain tissue that you see on the inside. And those different contrasts, how those tissues look, tell us a lot about the health of the brain. Okay, so first up I'd like to talk, I've already mentioned this a few times, so I've mentioned MRI, or magnetic resonance imaging. This is an imaging technique which uses very powerful magnets and radio frequency waves to create detailed images of the body, and in my case, the brain. Right? And so one of the many benefits of MRI is that it's non-invasive. What we mean by that is that it's non-damaging. It's very, very safe. And so people use MRI in many instances because it is quite safe, which is opposed to X-rays. So we're not using X-rays here. We're just using radio frequency, which is essentially the same thing that you can pick up in your car radio, which does no damage. But I would suggest that the most exciting thing about MRI is its flexibility. Right? And what I mean by that is that by slightly adjusting initial parameters, we can create a whole host of different types of images. And these different types of images can be used to look at the structure of the brain, or blood flow, or function, a whole raft of different things that are happening in the brain. So for example, if you go for an X-ray, you will get an X-ray. And that can be very useful. It can tell you about your bones. But when you go for an MRI scan, you will go for multiple types of scans. You'll be lying on the couch, the text will adjust a few little things, and the first scan will tell you about structure of the brain. And the second scan will tell you how much blood flow is in your brain. And the third will tell you about chemical concentrations in the brain. So it's incredibly powerful in its flexibility, as I think one of the main benefits of MRI. So, what I'd like to do from here is go through now some examples of images in brains, in actual brains, not fruit, and tell you about what we can actually measure. But first up, I did mention that MRI uses powerful magnets, and I really do mean powerful magnets. So what you can see here is a junkyard magnet, yes, and I assume that we would all agree this is a very powerful magnet. Yes, it can. When you turn it on, it can pick up lots of junk. It can pick up cars. So my very leading question to you is this stronger or weaker than the magnet that we use in MRI?
Weaken.
It is. It was an incredibly leading question. Exactly. So this is incredibly powerful. This is about 1 Tesla. That's how we measure the strength of the magnetic field. And the MRI that I use is 3 Tesla. Right? So 3 times stronger. And so that's one of the main reasons why, if you ever go for an MRI, you will always have a safety screen. Right? And that's to make sure that you don't have any metal in your pockets or loose on your body. So and the reason for that is that the magnet is always on. So whenever you go for that scan, you will need to go through that safety screen. So you can see 2 very dangerous examples here. No one was hurt in these 2 examples, but there have been at least now 2 documented cases where, for example, a cylinder has crushed an individual. So hence why that safety screening is important. But if we follow all those rules, MR can be very safe. Okay, one further thing. Now we're going to look at lots of brain images. And I'm going to walk you through how cool MR is and what it can tell us about the brain. But to do that, we'll just run through how we display these images. So here is a 3-dimensional image of the brain. Right? So what you can see here is here's our spinal cord coming up. This is what we call a cerebellum. This is at the back of the brain. And then you can see front of the brain, back of the brain. Okay? Sometimes it's difficult to display 3-dimensionally on a 2D screen. So what we tend to do is we will slice the brain in different directions. So here, what we've done is we've just sliced the brain horizontally. And what you can see here is that we're looking down on the brain from above. So here you can see the front of the brain, back of the brain, and the sides of the brain. So we can slice this a different way. We can slice it along the front, as if you were looking straight into the face of the individual, and that's what that looks like. So left of the brain, right of the brain, up and down. And lastly, we can also slice this, what we call sagittally. So that's when we make a slice straight down the middle of the brain, and we look from the ear, for example. And that's what we can see here. So here is, again, spinal cord coming up, front of the brain, back of the brain, and you're looking right down the center of the brain. Okay, hopefully that makes sense. And you can see what we're looking at when we slice the brain in those different ways. Okay, so first up, we have structural MRI. And this is kind of our workhorse that we use in almost every type of research scan or research session that we're doing. And this will tell us about the structure of the brain. We can extract really useful information about the volume of particular structures. We can look at the thickness of particular structures. We can even look at surface area, how much surface area we see in the brain. And this is an incredibly robust, very reliable measure that we see across a wide variety of research and in clinical practice. And so here we're just taking those three examples of the different views. So here we have a sagittal view. So this is looking at the brain this way. This is looking face on. And what you can see here, this is looking down from above. And so one of the things that I'd like to point out here is, again, getting back to that idea of contrast in the brain. So if you look here, you can see slightly grayer, darker areas in what we call the cortex of the brain, and that's the beautiful foldy bits of the brain where a lot of the processing and the thinking happens. And you can see that nice gray tones. If you look just below that, we see a little bit brighter, and this is what we call the white matter. And the white matter you can think of as the connections between those different gray matter regions, right? So if we have some thinking happening up here, but we also need to integrate and use this area of the brain, we have white matter, which is connecting those two areas of the brain. And lastly, if you look here, see these dark areas, this is what we call the ventricles of the brain. So these are fluid-filled holes in the center of the brain, which are filled with a fluid called cerebrospinal fluid. And this fluid bathes and protects the brain and bathes the brain. So you can see some beautiful detail there, and that gives us lovely, robust, reliable information about the structure of the brain. Okay, now we're not, as I mentioned a few times now, we acquire these three-dimensional images. So we're not limited to displaying just these slices. What we can do is create videos, right? So I can take all the slices and I can bunch them together and I can create a video which looks something like this. So I'll play this twice just to orient you. The first time we're going to look sagittally, so it's basically we're coming in, looking from the ear, and we're going to move this way through the brain. So see if you can identify a few things like the nose, the eyes, and obviously take a look at the brain. Because it's beautiful. Okay. So we're coming in to the brain now. We can see some brain there. Look for the eye here. Here's the mouth, the nose, and the different structures within the brain. And now we're moving across to the other eye and out the others.
Excuse me.
Yeah.
Just a question. Are we allowed to ask questions while you're talking?
Absolutely, please do. Yeah.
Right, okay.
Yeah.
Just wasn't sure if we had to wait till the end.
Do you have a question?
Well, going back, I just
Yeah.
wondered whether in those pictures just before where our lesions showed, where those ones here?
In this one?
Yeah.
Well, it depends on what type of lesion and whether that lesion would happen in the gray matter or the white matter.
Right.
And so sometimes you can see different types of images here, or different types of lesions. Here, for example, you can see tumors here on this type of image. But it depends on the type of lesion and the type of image that you're using to try and look at that lesion.
Right.
Whether you'd see it on this particular scan or a different scan, for example. Yeah. Right. Okay. So we can also just create these nice videos of slices as we move through the brain. But we can also then create these nice three-dimensional models. So this is what I've done here. This is my brain, and I've created this model. What I've done is to just slice down an edge of my brain so you can see into that big, beautiful brain, which is there. You can see these lovely folds of the cortex and the white matter. There's a few things that you can see. I'm going to start to rotate this around, and a few things to take note of. One, as you can see, I've had my eyes closed throughout the scan, and as you rotate around, you'll see there's no signal from my hair, which is normal. We don't actually see any signal from hair in MR. And as I rotate around the bottom, you'll actually be able to see some of the blood vessels coming up into the brain and feeding the brain with blood. So if I start this rotating, you'll be able to see those things. You can see my eyes are closed, you can see some nice definition on the ear, and as you can see here, a few vessels coming up from the neck, feeding the brain with blood, and then we're back around this is lovely, prominent brow full of just space. Right. Yeah, so please do ask questions if you do happen to have questions. I'm very happy to answer those during the talk. So, as I mentioned, MR is incredibly flexible. It's not just about structure. We can also look at things like blood flow. So we can tune the scanner to be specific and sensitive to blood, for example, moving in the blood vessels. So this is slightly difficult to orient yourself, but I'll tell you what we're looking at. So this is as if you're looking at me from the front, and what you're seeing are the two big blood vessels which are coming up your neck, the carotid arteries, which bring most of the blood up into your brain. And we have this one tiny one at the back, the vascular. Just focus on these two here. And so we can actually look at how much blood is coming up those vessels and entering the brain. So for example, I have a little video here of modeling the blood flow as that moves up the vessels into the brain. And this, for me, is incredibly beautiful because Ashley, my PhD student, spent months making this image. It may not mean that much to you, but it's beautiful. So it's beautiful because you can actually see we model how the blood flow comes up and then bends around the curves and then splits as it enters the rest of the brain. And we can measure how much flow we're seeing, how elastic the vessels are, how the vessels respond to pulsatility from the heart, and we can see how healthy the vessels are in this case. We can continue to follow that blood flow up into the vessels and then see how it perfuses or how it actually feeds the tissue with that oxygenated blood full of nutrients that all of our tissue needs to survive. And that's what we're seeing here. So what we're seeing is a series of horizontal slices. So looking down on the brain from above, this is at the bottom of the brain, moving up towards the top of the brain on the other side. And what you can see here in red is where we see areas of high blood flow arriving at the tissue. And areas of blue show slightly lower levels. And this is what we would expect. So around the edges of the brain, we have that gray matter, where a lot of the thinking and processing happens, which we therefore need a lot more blood there to bring the oxygen and nutrients to allow us to perform all that metabolism. And there's actually less necessity for, well, we need blood obviously in the white matter, but we don't need quite as much. So you can see a difference there. Now we can actually continue, and actually maybe getting back to one of the question about lesions here, we can actually continue that forward, and in some cases you can start to get maybe even some small blockages in these tiny little vessels. And if that happens, we can see that showing up as something which we call white matter hyperintensities or white matter lesions. And these are small little lesions which you can see occurring of a vascular origin, so that probably has something to do with blood flow. And you can see these, maybe not that clearly with this lighting, but this is a different type of scan, not the one that I showed earlier, and there we do see lesions showing up on this particular scan. So what you can see here is, hopefully you can see a little bit of whitish or brighter signal in these areas, and we have some fancy algorithms which we can use to identify those. And these are what we call white matter lesions. So this would be an example of one type of scan which we can use to identify both the location and the extent of specific types of brain lesions. Right. Now previously I mentioned that white matter. The white matter which helps us connect one region to another region. And we can use MR to actually map out those structural connections and create rainbow pretty pictures as well. So what we're looking at here is what we call a tractogram. So this is a specific type of MRI, a diffusion MRI, which allows us to look at the health and structure of the white matter, the connecting tissue between different regions of the brain. But one of the cool things that we can do is we actually can identify the direction that these different white matter pathways are running and connecting. So in this image, it's what we call a coronal view, so that's looking face on to this individual. And And what we can see here is blue is indicative of fibers which are moving in the up-down direction, right? So here we can see the spinal cord coming up, and we can see these fibers which move up through the brain and then up to the top of the brain, which we call the motor cortex. You can also see this beautiful red fiber. This is called the corpus callosum. This is a big white matter fiber which connects the left and the right hemispheres. And we can see these two green bits, which are probably my favorite white matter pathway in the brain, called the cingulin bundle. And these are two, a right and a left, which run from the front of the brain to the back of the brain. Incredibly important for transferring information from the front of the brain to the back of the brain. And so you can see these not only the location, but you can also see the direction that they are connecting, and you can look at metrics of health along those metrics to tell us anything about aging or disease or development. But again, we're not entirely limited to structure. We can also look at function. And so you've probably heard of functional MRI or fMRI, and this is an example of two types of functional MRI. So what we have here on the left, this is what we call a resting state functional MRI scanner. And so what we've done here is we've put the participant down on the scanner, we've put them into the scanner, we've turned on the functional MRI, and we've asked them to lie there and do nothing. Right? So we've asked them to just not think of anything in particular, do not perform any task, just lie there. And what you're seeing here in yellow are the random activations. So we're seeing areas in the brain which are constantly increasing their activity and decreasing their activity. Not in response to anything external, it's just the brain doing its thing. And what we can see from this, we can apply some fancy mathematics to this, and we can pull out what we call functional resting state networks, which is specific areas of the brain which are more likely to communicate with other areas of the brain. So for example, we have one which is called the default mode network. And this is, you have a big node here at the back of your brain and a node at the front of your brain, and these two areas are highly connected functionally. And they're much more connected to each other than they are to other areas of the brain. And this can tell us, the state of these networks can tell us about, again, health, disease, aging, and can be very informative. The more common usage of functional MRI would be something that you see here on the right. And this is the back of the brain. What we've done here is we put the individual into the scanner, and we've put some, they can see a screen, and on the screen we put up a really annoying flashing checkerboard. And we get the people to look at this flashing checkerboard, and then we'll turn this checkerboard off. And then we'll look at the flashing checkerboard, and it'll go off, and it'll go on, and it'll go off, and it'll go on, and it'll go off. And it unfortunately takes a little bit longer than you would potentially like to look at a flashing checkerboard. But people are very, very generous with their time and their effort. So what you can see there is, then we look around the brain and we say, right, where in the brain do we see this nice pattern of increased activity with the flashing checkerboard, and then decreased activity when they're not looking at the flashing checkerboard, and then increased and decreased and increased and decreased. And what we can do is we can identify this area in orange and red, which is at the back of the brain, which is what we call the visual cortex. Right? And the visual cortex is responsible for responding to visual stimuli. So that flashing checkerboard, we see an increase in activity at the back of the brain, and we can pick that up. The other thing to note is that this is completely non-invasive. Right? We haven't injected anything. We're just taking a picture of how the brain reacts to an external stimuli. And so, if we extend from that, we can do things much more clever than just asking someone to look at a flashing checkerboard. We can start to ask interesting questions about maths or ethical dilemmas or what have you. You can come up with some fairly interesting questions, would be my guess, and we can try and look at where in the brain we're seeing these activities and how is the brain trying to process these different questions. So it's a really powerful and interesting technique. Right, and there are many, many others, right? Many, many other types of MR, and clever people all over the world are coming up with new ways to use MR to look at new measures all the time. It really is a fascinating technique. On the one hand, we have some really developed techniques, which we can use in the clinic every day, but on the other hand, it is still evolving every single day. So it's really exciting. Okay, do you guys want to hear about positron emission tomography?
Yes, please.
All right. Well, I was going to skip it, but I can't. I'll do this. It is does have a special place in their heart. So, switching gears just slightly, right? Switching from MRI to another type of brain imaging, which is called positron emission tomography, or PET imaging. So this is a completely different machine and works in a completely different way. And the way that PET imaging works is that you need to inject radioactive material into the body. And you need to get your chemistry right. You put this radioactive material onto a specific chemical that you're interested in, and then you inject that into the body. This chemical flows around until it binds to the particular chemical in your body that you're interested in, and then radiation comes out of your body, and we use this circle detector to identify where the radiation came from, and then we can create, again with some fancy maths, a beautiful picture of where the radiation came from. And if that radiation was tagged to glucose, for example, which will tell us about metabolism, so we can look at metabolism in the brain, or if that radioactive material was tagged to a protein like amyloid, which you may have heard of as one of the pathological hallmarks of Alzheimer's disease. We can also look at how much amyloid we actually see in the brain. And we can, again, it's very flexible. And I would say that, again, if you get your chemistry right, you can look at metabolism, neurotransmitter function, so you can look at dopamine function, or serotonin function, or a whole host of different things. So again, it's incredibly powerful and very flexible. Now, the reason that it's interesting to me and to us at the New Zealand Brain Research Institute is because of the new ability to look at protein deposition. So, as I alluded to earlier, so you probably have heard of amyloid protein. This is, again, a hallmark of Alzheimer's disease. This is a protein that we have, all of us have in our brains and in our bodies, and in some pathological process, we don't actually know how that happens just yet or why it starts. The amyloid starts to misfold and it clumps together and it gets deposited in the brain. And this is one of the, again, one of the hallmarks of Alzheimer's disease. And so what we can do now with PET imaging is we can actually image how much amyloid has been deposited or accumulated in the brain. And again, completely in vivo, right? So we can do this here in Christchurch. And I have an example of what we call amyloid negative. So this is an individual who we've scanned with this amyloid PET technique. And this is what a negative scan looks like. That means that we're not seeing very much amyloid deposited, specifically in the cortex, around the edges of the brain. But over here, hopefully you can see the difference. That's what we call amyloid positive. Right? And so that individual was showing quite substantial accumulation of this Alzheimer protein in the brain. And so this can be used both in research, but is starting to be used more and more clinically, specifically around young individuals, where there's a real question about, is this Alzheimer's disease or is this something else? And specifically using this to say, if we're seeing amyloid, most likely on an Alzheimer trajectory, and if we're not seeing amyloid, probably not on an Alzheimer trajectory. But again, I'm very happy to answer questions about PET imaging, but there's a little taster into PET imaging and some of the PET imaging that we're doing here.
So could you pick up if somebody had Alzheimer's before they actually started showing you?
That's a very good question. So what we can see is that we know that the deposition of amyloid is associated with age as well. So when you look at individuals who are 60 to 70 years old, about a quarter of those individuals will have an amyloid positive scan. The question that you're asking is, well, is that just early Alzheimer's disease before we've actually seen any further consequences, before any memory loss, for example? Or is it just amyloid deposition? And we don't know the answer to that question yet. So, for example, there was a really famous, they call it the NUN study, not N-O-N-E, but N-U-N, of a group of nuns who were very old, right? They all made it to very close to 100 years old. And they came to autopsy, and we looked at their brains, and they were full of amyloid, and they were perfectly fine. Right? They had no signs, no symptoms of Alzheimer's disease. So we do know that if someone has an amyloid positive scan, they are at higher risk for developing Alzheimer's in the future. But it's not a 100% sentence. There are still lots of individuals who will be living their lives very happily with lots of amyloid in the brain with no symptoms whatsoever. So it's still an open question as to the actual meaning of a healthy individual showing amyloid positivity. Good question. Okay. Excuse me. Right. So, what I wanted to do now was tell you how we're using some of these techniques at the New Zealand Brain Research Institute across a wide range of different research topics. So, at NZBRI, we use imaging to try and answer what we think are interesting questions around Parkinson's, Alzheimer's disease, including some questions about multiple sclerosis. But we can move then to the other end of the age spectrum where we look at child development, very low birth weight, preterm birth, and how we see development there, and then a lot of other unrelated, or sometimes related, questions in between. So from diseases of the elderly to child development and everything in between. So one of our main interests is Parkinson's disease, and specifically around potential cognitive difficulties or memory, thinking, potential difficulties that we see in Parkinson's. And can we use brain images to tell us anything about this? But before I tell you about my area, which is brain imaging, I will just mention that at the New Zealand Brain Research Institute, we have quite a large Parkinson's research team, which spans not just imaging, but looks at eye movements, bloods, so plasma biomarkers, so drawing blood and seeing what we can see from there, EEG, looking at motor and genetics, epidemiology, so a whole raft of can we understand what's happening in Parkinson's a little bit better. And one of those pieces of the puzzle is brain imaging. And so the way that we've attempted to answer this is with the New Zealand Parkinson's Progression Program, which is a mouthful, so we sometimes call it the NZP3. And so this has been going almost 15 years now, started in the late 2007, and has, some of you may be familiar with this study and have maybe even participated. But we have a large number of Parkinson's patients now who have entered in this study and have gone through a wide range of different assessments, from cognitive tests to motor tests to behavioral assessment to clinical assessment. We take bloods from them, we look at MRI, and we've even now started to look at PET imaging from these individuals. And so, a quick summary of some of the MR findings that we found. Maybe not the most eloquent, but you know what I mean. So we do have some individuals with Parkinson's disease and dementia. And from these individuals, what we've seen is, as a group, I'm displaying three different types of MR here. So here on the left, I'm showing, on average, what do we see happening in individuals with Parkinson's disease and dementia? And in terms of tissue loss, do we see any tissue loss? Well, we do see some tissue loss. Areas that you see here in blue and green are indicative of areas that are showing less tissue in individuals with Parkinson's disease and dementia relative to healthy individuals. Now, we can also ask, do we see a similar level of changes in blood flow? So what you're looking at here is blood flow or perfusion. And what you see in blue and green is slightly more widespread, right? So we're seeing actual functional loss of blood flow in these cortical areas relative to the slightly more restricted areas of tissue loss in Parkinson's or dementia. And lastly, if we look at white matter damage, so now this is displayed slightly differently. We're just looking at the white matter here, remembering the connections. And what we can see there is in areas of red, we're starting to see some loss of integrity. So slight damage to the white matter occurring in Parkinson's disease and dementia relative to healthy individuals. Now, this is obviously Parkinson's with dementia. When we think of other groups of individuals with Parkinson's, so for example, those who are still functioning normally, cognitively normal, living quite well, we don't see these differences. And we are not able to identify any significant differences relative to controls. So, a quick summary of a bunch of different types of imaging metrics. In our Parkinson's work, we're now looking at this longitudinally. Can we see changes over time? Can we use these images to predict outcomes or predict how the trajectory of an individual? And what we're seeing is we have a few images which are not helpful at all. This is interesting, right, because they don't tell us anything about the disease, and we have essentially eliminated these from any of our future work. It saves time, it saves money, and obviously it saves participants slightly uncomfortable time in the scanner. We do have some images which are showing some information, some useful information about the progression of Parkinson's disease, and we have some new scans which we really don't know yet, right, and we're still looking at those. But I think these will tell us about some interesting aspects of Parkinson's. So that was just a quick summary of some of our Parkinson's work. But I would just like to illustrate how the different images can be used across a number of different research areas. And one of those areas that more and more people are interested in is aging, general aging, right? And so we have two incredibly wonderful studies here in New Zealand. You may have heard of the Dunedin study, the multidisciplinary health and development study in Dunedin. And so this is a study of individuals who were born in 1972 and have been followed almost 20 different times. So we're coming up, it was in Dunedin last week, we were setting up the MR scans for the age 52 assessments. We'll start those a little bit early, but they're not 52 just yet, but they're almost 52, and we'll be following those individuals through. The Christchurch Health and Development Study is a similar study. So these were for individuals born in Canterbury in 1977, and the same type of thing, how they've been followed over time. And the Dunedin study and the Christchurch Health and Development Study have both added brain imaging as an assessment. They have added at age 45 and now again at age 52. And so now we're really being able to start from, the study was really designed as a development study, as how does early childhood events affect later life, and now we're starting to move into questions of aging. So it's a really interesting time, and again, a really useful and impactful application of brain imaging. So we found some really interesting things, but we're really excited for what the future holds in those particular studies. So sleep apnea, I'm sure that most of you have heard of sleep apnea. This is a condition where individuals will essentially stop breathing in the middle of their sleeping time, where you can have disrupted sleep, reduction in oxygen to the brain. And so some of our colleagues have looked at blood flow MRI of the brain. And these areas of blue that you see are areas that are showing lower levels of blood flow in individuals with sleep apnea relative to individuals who don't have sleep apnea. And the really exciting thing here is that they've moved on to a treatment trial, which is looking at not severe sleep apnea, but mild to moderate sleep apnea, and using CPAP, which is the positive pressure, which is the treatment for CPAP, and looking to see if that actually makes a difference to blood flow and other measures of sleep, motor health, and cognitive health. And while that study is ongoing, it's looking promising. So potentially something there and a nice outcome that we can use MR to assess how these treatments are working. We've also done some work looking at chronic pain. We all know that chronic pain can be quite an issue for essentially all members of society. And what we did here was we actually found some really interesting things. In a group with chronic pain, we found smaller levels of tissue, smaller gray matter volumes in the cerebellum, which is this area here. And that's really interesting because we know that the cerebellum is involved in in a lot of motor coordination. And a lot of these individuals with chronic pain were obviously experiencing this pain during motion and movement. So it's quite interesting. This was a novel finding. We're still trying to parse out what exactly it means, but some interesting findings as well. Now, lastly, I'd like to tell you about two different studies which are independent but related. And so this is looking at development. So this is looking at a group of individuals who were born very preterm. So they were born under 28 weeks gestation. If you remember, the full gestation is about 40 weeks. So these are quite preterm. They've been followed a few times until up to 12 years of age when we scanned them at age 12. And what we found was really quite interesting. So here in areas of blue, you're seeing not quite as much tissue, so smaller levels of tissue in the preterm birth. But here in red, you're seeing actually higher levels of gray matter void, which is quite an interesting question. So the questions then arise of, are we seeing differences in these connections? Sometimes these differences in atrophy are related to attentional differences. We're not quite sure, but the really interesting thing is that we looked at a group of individuals who were born with very low birth weight, so similar to, but not exactly the same as preterm birth, and we imaged them at 27 years of age. And what we were able to do was essentially recreate those findings, showing smaller levels of tissue in the blue areas, higher levels in the red areas. So what does this mean? It's a very good question. I can't actually pin down into exactly what this means, but the interesting thing is that it's reproducible across two individual cohorts with preterm birth and very low birth weight. So we're sorting this out at the moment, so we'll watch this page, and it's quite interesting. Again, a nice application of MR. Now, the penultimate thing that I'd like to tell you about are the dementia prevention research clinics. So this is a nationwide consortia or nationwide emphasis looking at describing, understanding dementia, moving towards prevention of dementia. So developing a big group of individuals who we can then test out new interventions and new therapies. And this is, imaging is one of the fundamental metrics that we're using to assess individuals. As I said, this is nationwide, so we're recruiting individuals in Auckland, Christchurch, and Dunedin. And this is well into its about 4th or 5th year. And we're, again, starting to find some interesting changes in brain scans in individuals that are looking like they're trending towards dementia. So, I lied. The second, again, this is going to be penultimate again, but the second to last thing I'd like to tell you about is up on the screen now. So again, I give you a question. This is a human brain. This is not a human brain. Does anyone have any idea what that brain belongs to?
Frog.
Oh, sorry,
Frog.
frog, not a frog? No. I'll give you a hint, it's a mammal.
Sheep.
It's a sheep. It is.
He's a farmer.
Very well done. Very well done. I mean, I was just as an aside, I was shocked when I saw sheep brain for the first time, and that it's incredibly similar to the human brain, right? I mean, yeah, this size is slightly different, and the shape is just a little bit different, but we have very similar gray matter, white matter, ventricles, and essentially we image them the exact same way. We do have to knock the sheep out. We do a little bit of anesthetic, but they're fine. They come back. They're fine. But I bring this up because this, I think, is a fascinating study. So this is a study that I feel really fortunate to be involved with. This is run out of Lincoln, and they're looking at this pediatric neurodegenerative disease called Batten disease. So it's a rare disease, and it's genetic disease. And what happens is kids will develop normally until 2 or 3 years, and then they will start to have motor problems. They'll lose their sight, and they will then eventually die very, very young. So it's an atrocious disease. It's a horrible disease. However, we know that it's caused by a series of genetic mutations. Now, these same genetic mutations were identified in two different, in a merino and a South Hampshire, Borderdale, excuse me, merino and a Borderdale sheep here in New Zealand, and those two herds have been maintained out of Lincoln, and over the past 25 years, they've done an incredible amount of work looking at treatments towards this disease. And we've come up to the point where we have a beautiful gene therapy that they've developed that we can now inject into the brains of these sheep, and we're seeing incredible results. And I just bring this up, and I'm going to show you one plot, just to sneak in one plot, about how amazing this treatment is. So we've used MR to help track how well that treatment's working. So here's just a visual, right? So we've scanned these sheep at 5 months, 7 months, 10 months, 14 months, 18 months, right? So we scanned them 5 times over a year and a half. Here's a healthy sheep. And what you can see is a healthy sheep basically just stays the same over that time, right? We don't really see any changes in size. What we see is an untreated sheep who's affected. And what you can see is if you look at the size of the ventricles here versus the size of the ventricles there, you can see that the ventricles are getting bigger. We're losing tissue over time. And here is a treated sheep. And what you can see, again, is that with the treatment, we're not really seeing any change in size in the brains of these sheep. So if I would put that into one plot that I've included in this presentation, because I think it's so cool. So here, these are our controls, and this is total gray matter, how much gray matter we see in the brain. You can basically see that over time, the controls are actually getting bigger, they're growing over time. The treated sheep, they're slightly lower because we didn't treat them until slightly, a few months into their life. They are maintaining, they're not getting worse, right? And these are the affected sheep where we didn't treat, and you can see they just fall off the cliff. Right? And so this is incredibly exciting. And a really nice example of a really great collaboration from using non-human animals and gene therapy and brain imaging, putting it all together. And this, there was a clinical trial which started in the States in mid last year based on these results. So that's already been tested in 2 individuals now, 2 kids in the States. So very exciting, and we'll see how we go there. But before I do that, I'm going to leave you with one last thing, and I'll leave you on a positive. And this is one of my favorite studies. This is an old study now. 2004 was the first time they did it, but they've repeated it multiple times. And this is the juggling study. And all brain imaging people know the juggling study. So this was originally performed in university students. So they got a big group of university students together, and they scanned all of them. And then they separated them into 2 groups. And the first group, they said, just live your life normally, don't do anything different, just keep doing what you're doing. But in the other group, they gave them juggling training. Right? So they instructed them on how to juggle, and they practiced together, and they worked through it, and they got to a point where they could maintain the 3-ball cascade, right, which is just 3 balls, and they could maintain that for a minute. And when they could do that, they scanned everybody. Again, both groups. And what they found was that in these areas of yellow, they saw increased gray matter volume. They found more gray matter tissue in the people who learned to juggle. Right? So the press got a hold of this, and it was like, oh, we found the areas of juggling in the brain. Obviously, the brain doesn't work. Right? There's no juggling areas in the brain. The point of this study, which is so exciting, was that they were able to show a structural change to the brain as a consequence of learning something new. Right? And that's why it's so exciting. And it just goes back to highlight that if you stay mentally active, physically active, socially active, and have a healthy diet, those are all good things for you, right? And this was just the first example of how we can use MR to show a structural change in the brain as a consequence of learning something. So, it's not all doom and gloom, and we can still do things to help our brains. So, with that, I will stop, and I'll say thank you very much. A lot of the stuff that you've seen here has been collaboration with amazing colleagues at the New Zealand Brain Research Institute. And thank you very much for your time and for your attention.
Thank you. Thank you so much.
Well, I'm certainly happy to take some questions if anybody has any questions. Yeah.
Yeah. Do Parkinson patients anyway, they when this kind of getting into the sort of getting worse, do they really get the dementia?
Do they really get dementia?
Yeah.
Or?
Yeah.
So we do see a higher percentage of people with Parkinson's disease developing dementia than the general population. But it's certainly, again, it's not a certainty. Right? If you were diagnosed with Parkinson's disease, there is not a certainty that you will develop dementia or not develop dementia. So and that is one of the tricky things that we still don't know the answer to, and is something that we are certainly trying to work towards, is to try and help identify who is at risk of dementia in the near future and who is not at risk of dementia in the future. The idea being there, then if we can identify those who are at high risk, which we can't yet, but if we can identify those who are at high risk, we can then preferentially slot them into new treatment trials try and prevent that dementia before it converts into overt dementia. So that's that's a lot of what we think about, is how can we help identify those individuals who are at risk to then try and target treatments to them. But we're not there yet, unfortunately.
And how are we gonna know that
How are we going to know?
that we're developing that dementia?
How are we going to know that we're the
patient
individual is
anyway.
developing dementia?
Yeah, the patient anyway.
So one of the ways that we can help, or one of the ways that we can identify dementia at this point is still a clinical, clinical diagnosis. So the way that we identify that is with our cognitive tests, our neuropsychological tests. And so we do a lot of those tests. We compare those to healthy individuals who don't have Parkinson's, for example. And then there will be specific cutoffs, and we say, well, we're going to set our line here, and we're going to talk to the clinician, and we're going to talk to their significant other, and we're going to make a subjective, because we do have some numbers, but it is a subjective decision as to when someone is classified as dementia or not. Yeah. So there are ways that we have agreed that we identify dementia, but we still don't. If someone comes to me today and said, I was just diagnosed with Parkinson's, am I going to get dementia? I can say, I can't tell you I can't tell you if you will or you will not. And that's a problem. We're trying to answer that question with a lot of what we do at Canterbury.
Thank you.
Yeah. Yeah.
Hi. I was diagnosed with Parkinson's in September last year, and in doing so, the neurologist showed me a scan of my brain, and it showed that the left hemisphere of the brain was dropping off from dopamine cells.
Yep.
And so I didn't see any of that today, and I just sort of wondered, is that different or?
Yes.
I'm still trying to understand what it's all about.
Yep. So what you would have seen would have been a dopaminergic scan or a dopamine scan. And you can look at that in one of two ways. You can look at that with PET imaging, or you can look at that with SPECT imaging. It's another type of imaging. The SPECT imaging is what we currently have in the hospital system here. And that is what you would have seen. When you look, it's really grainy. It'll be really grainy.
Quite colourful.
Yeah. Yep. So usually it's pretty grainy, and we color code it. And generally you will see an asymmetry. So you can see one side looks smaller than the other side. And that is one of the confirmatory ways that we can identify Parkinson's. So I haven't talked about any of that here.
No.
But that A does exist, and B can be very helpful especially in differential diagnosis. If you have an idea that there's something Parkinsonian happening, but you're not sure if it's going to be something like Parkinson's or essential tremor or a few other things, one of those scans can be particularly helpful in sorting that out. And the reason that we see that asymmetry is that generally we see a unilateral or one-sided onset of motor symptoms, and that corresponds to the dopamine-producing cells on one side of the brain as well. Yeah. So that A exists, B is accessible here in Christchurch, and C can be very helpful. Yeah.
Yeah, I was just wondering, can you see in an MRI scan when somebody's heart rate is elevated, can you see, measure the blood flow to the brain?
That's a very good question. I think about that a lot. And I think I have three answers for you. So, one, on structural scans, you don't really see that because you're not looking at that. However, it can have an effect because if people have an elevated heart rate, maybe feeling a bit anxious, that actually can translate to a bit more motion. And just like when you take a snapshot, if people are moving very quickly, it gets blurry. Same thing happens in MR. If you get motion in the MR, it looks a bit blurry. So that's not actually your question, but it's something I think about a lot, trying to get really nice images. Now, if you are measuring blood flow per se, if you are looking at flow coming in through the vessels, we can definitely pick that up. We can definitely pick up changes in pressure, changes in flow, as a consequence of anxiety or potential drugs someone's taking, or We actually do see it, when I don't know if this happens to any of you, it certainly happens to me, when I'm sitting or lying down and then I stand up and you feel a bit dizzy, and we know that we see those changes in pressures and flow. You can actually pick those up if you're trying to pick those up, you can pick those up and flow a bit. And third answer, I can't remember what it was. But I can answer your question.
Well, that's supplementary.
Supplementary question.
Does it do the brain any good to intentionally increase that flow by, for example, recently strenuous exercise?
Ooh, I definitely have some good answers to that one. So there's a few things there. One is we have this system of autoregulation, is what we call it, which is basically your body and your vessels want to keep sufficient amount of blood going to the areas of tissue that you need it to go. And so that's an example of autoregulation when you go from lying down to standing up. If it doesn't work immediately, you feel a little bit woozy until it kicks in, and you will adjust the flows and the pressures and the stiffness and get the blood up to where it needs to go. One of the things that we have been thinking about is identifying areas of low flow. Can we increase those levels? Can you increase blood flow? There's been some interesting work looking at a particular family of drugs to actually increase that blood flow. I know that there's been one clinical trial which was not quite as exciting as we thought it would be, but there's still some ongoing ones looking at cholinesterase inhibitors, for example, to try and increase blood flow pharmacologically. And then the third is, so for example, strenuous exercise. What I can tell you is that as a blanket statement, what's good for your heart is good for your brain. So increased physical activity and maintenance of that physical activity, not only is it good for your heart, but it's certainly good for your brain, and specifically around blood flow in the brain. Yeah.
Sorry, you probably answered the question when you answered the first question about with people with MS, are they at a higher risk, like Parkinson's, they're getting dementia?
I don't know enough to give you an informative response to that. I know that there's been a lot of work, and I've worked with people who've been on spectrum C40, but I don't know the answer to that. I can't give you a definitive yes or no. Sorry on that. But I do know that there's been a lot of work looking at what does happen to cognition in individuals with MS, but I don't remember the outcome, and I'm not steeped in the literature enough to know.
Now, a study would you call that might, if someone
Oh,
children?
yeah. I mean, I think if you look for something like risk of dementia in MS, although that's always different, that's always dangerous. That's always dangerous. The way so much is blown out of perspective, and blown out of proportions.
Because would there be somebody on your team that would come and talk to us like you have done today?
Oh, absolutely, yeah. So we have, for example, Dr. Debbie Mason is a neurologist here who specializes in MS, and Ty can pass on her information. She is a fountain of knowledge.
Yeah, I know. It's probably mostly
I just thought that if Dr. Mason could come and talk to you, that'd be great.
Yeah, I'll give her details.
I'll let her know you said yes.
She would love to talk. She will make space in her schedule. Yes. And I have touched on a few things today, and there are a lot of really excellent researchers and clinicians at NWRI who I have already suggested that they need to come and talk here. So I did sign them up. Yes.
Do you know of any research that's being done in New Zealand on diet and its impact on neurological disease? I'm thinking actually about the keto diet and and the effect of ketosis on the brain. Is that something that imaging could capture?
great question. So sorry, I haven't been repeating the questions. Did everyone hear those questions?
Right.
Sorry. Yeah, so this was a question about if there's any research here in New Zealand on diet and neurological conditions, and specifically around the ketogenic diet. So I do know, Matt, what's Matt's last name?
From Hamilton?
Yes.
Yeah.
Yeah.
Who was in America?
Yes.
Yeah.
Yeah. I can't remember his last name.
Phillips, perhaps?
What was that?
Phillips?
Yes. Yep. Yeah. So he's a neurologist. He's in Hamilton, isn't he? Yeah. Neurologist up in Hamilton who has done some really interesting work. I think it's really interesting. And he's really thoughtful about how do you set up a study to actually test the effects of ketogenic diet versus not just any other diet, but some diet which has been matched in macronutrient profiles. So I'm thinking about the relationship between carbohydrates and proteins and fats and so his first studies were really, really thoughtful and really interesting. I think what would be really, that was exactly what I was going to say, would be really nice to see that in a much larger cohort. Right? It would be really nice to follow that up and see, can we see that effect flowing through across all individuals? Is it a subset of individuals? Yeah, I think those questions too.
How do people get to volunteer to, be party to, if anyone wanted to do a study like that? How is there a way of collecting up a group of individuals who might be interested in doing that?
Yes. So, yes. So, how does that work? So, there will be, drives, recruitment drives, when there's a specific study which has funding to do a specific thing, and there'll be advertisements, Facebook. A lot of the time, we now actually try and connect with other organizations which may have individuals who would be interested, and try and access interested individuals that way. A lot of the time, there is just an announcement which comes up on the specific university groups webpage or Facebook. So, if there were a centralized, here's neurological research in New Zealand, and there are 27 studies happening, and there's 2 in Parkinson's, and there's 4 in Alzheimer's disease, and they're specifically looking at that, would be really helpful, but no one is organized. But that would be a good thing for everyone, for potential participants, for researchers, for clinicians, for everyone. It's a good idea.
And he was interviewed on Kim Hill on national radio.
Yeah.
So you Google that and can listen to it.
Yeah.
Is there much research into the restless leg syndrome, which I understand is an offshoot of Parkinson's?
Mm-hmm. Yeah, so the question was about restless leg syndrome and this relationship to Parkinson's and whether there's any research looking into restless leg syndrome. So I can speak from NZBRI perspective. So it's certainly one of the things that we ask about. One of my colleagues, well, 2 of my colleagues, Tony Pitcher and Daniel Mile, who I'll also get to come talk to, because they're both wonderful and they're fascinating to listen to. So Tony is leading a nationwide study, which is an epidemiological, sorry, a tricky word to say, which is trying to look at environment, so life histories of individuals, where they were born, where they lived, exposure to job sites or chemicals, in addition to genetics, in addition to health information, trying to put that all together to try and sort out, can we see any links? And she's definitely asking about restless leg syndrome. So while it hasn't been a focus of the imaging stuff that we've done, it's certainly on the horizon. It's certainly part of ongoing studies from colleagues at the institute. Yeah.
Okay. I'm probably not the only one here who's been involved in that one with Tony Pitcher, but it's the depth. I've gone back 4 generations with questionnaires and all sorts of
it's
related
very
information.
thorough. Yeah. How did you find the? It wasn't too onerous. It wasn't too onerous.
No.
Good.
No.
Yeah. she's put a lot of thought into that. It's I
the
think
one-on-one online was quite challenging.
Yeah, absolutely. So I think that really does have the potential to be really New Zealand specific, right? And think about the unique New Zealand environment and exposures and what that could potentially mean for Parkinson's here. Yeah.
Just wondering about the results of the saliva test, where we get our DNA history as well.
Well, so you won't get the specific history. Like we don't look at it from like the ancestry perspective that you can from specific places online. We're more interested in specific potential markers of Parkinson's, the relationship between genetics and environment and Parkinson's. So we'll see. I mean, I think it's going to be a really, really nice study. It's really powerful to answer some really interesting questions. So on her behalf, I will say thank you, because I know she will be very appreciative.
Quite concerning in the latest neurological magazine that's just come out, and they say 1 in 3 New Zealanders will be affected somehow neurologically.
Yeah. I know the statistic 1 in 5, but it doesn't really matter, because 1 in 5 is very high as well.
Yeah.
Yeah. I think that just goes to illustrate that this is a major issue. Right? Neurological, I mean, the brain is incredibly complex, incredibly important, and lots of things can happen, right? Some of those things can be transient. Some of those things you could experience at some point in your life and then completely resolve. Some of those things are not. Some of those things will be lifelong. And I think it just goes to illustrate that this is an issue, and this is something we need to pay attention to, and both from a clinical perspective and a lifestyle perspective and a research perspective. And I think it is incredibly important. Right?
Thank you so much. Yeah, thank you so much.
Thank you for coming, Tracy.
Yeah.
Felt like I was back in my own country there. And I was actually really impressed about the knowledge of the breed of sheep. So I feel like you've integrated